Hey guys, today we're going to talk about genetic material and how it was discovered and the structure of DNA. First, let's look at the history of genetics and things. So remember that Mendel and other scientists figured out how inheritance patterns worked. So they figured out how X-linked traits are passed, how other types of traits that are polygenic or codominant traits are passed, but they did not know how things were inherited. So they needed to find out what is the genetic material that's passed from parents to offspring. Is it proteins or DNA? And there are several different scientists who contributed to discovering that DNA was in fact genetic material. One of the scientists who helped discover that DNA was genetic material was a scientist named Griffith. Griffith did an experiment where he injected two types of bacteria into mice. Some of the bacteria that he injected was virulent, which means that it causes disease, and that bacteria was called smooth bacteria. He also injected rough bacteria into different mice, and that is non-virulent, which means it does not cause a disease. He discovered through this experiment that the process of transformation occurs. Transformation is when a change in genotype is caused when a cell takes up foreign DNA. If you remember genotype from the genetics unit that we finished before this new trimester started, it's the genetic information that causes a physical trait in an organism. Now we can look at a summary of Griffith's experiment. So he took some bacteria, the non-virulent bacteria, and this was the bacteria that does not cause disease, and when he injected it into mice, the mice remained healthy. However, when he injected smooth bacteria into a mouse, which is virulent, when the mice were infected with that bacteria, they died. If he killed the smooth bacteria first by heating it up so that it was no longer alive, that heat-killed smooth virulent bacteria was no longer deadly to the mice, and the mice remained healthy. However, if he took rough, non-virulent bacteria that was alive and he mixed it with heat-killed smooth virulent bacteria, so normally this does not cause disease, and in this case, the DNA from the heat-killed bacteria transferred into the rough bacteria because when the mouse was injected, the mice died and produced live, smooth bacteria because the DNA from the rough bacteria was no longer expressed. Another scientist that assisted in helping determine the genetic material in living things is a guy named Oswald Avery. His experiment reinforced Griffith's conclusions. He did an experiment that broke down both proteins and DNA using enzymes. He discovered that transformation stopped if DNA was broken, but it did not stop if proteins were broken. Several years later, another scientist named Erwin Chargaff discovered that within a DNA molecule, while they didn't know the structure, they did know some of the chemicals that were inside of it. He discovered that the number of adenine molecules in DNA was always equal to the number of thymine molecules. And he discovered that the number of guanine molecules inside of DNA is always equal to the number of cytosine molecules. And that information is going to be really important later on when some other scientists are trying to figure out the shape of a DNA molecule. The people who finally convinced science that DNA is in fact the genetic material and not proteins were a duo of scientists named Hershey and Chase. And they did a pretty cool experiment where they used bacteriophages. A bacteriophage is a virus that only infects bacteria. And so they took a virus to infect a bacteria to prove that DNA was genetic material. Before we talk about their experiment, we need to know how a virus works. Now we know that a virus typically will land on the surface of the cell, and then most of the virus stays on the outside of the cell, but some of the virus is injected inside of the cell. The part that's injected inside the cell is DNA. The DNA from the virus will then tell the cell to start making virus parts. When it does that, the cell stops making its own essential parts and starts making all of these baby virus parts. And so then the cell becomes a factory essentially for viruses, and it assembles complete viruses. And then the cell will burst open and it will release all of these new viruses out into the world. So that's typically how a virus works. Here you can see what a cell looks like when it's infected with a virus. So this is a bacteria cell here. This is the surface of the cell. And then you can see these are all viruses that have landed on the surface of that cell to inject their DNA inside. You can also see in this picture, these are all new viruses that are being formed inside of the cell that are ready to go when the cell bursts open to release them. Now we can look at Hershey and Chase's experiment. So Hershey and Chase took two different phages, and they made one of the phages radioactive with phosphorus and the other radioactive with sulfur. Phosphorus is found in DNA and sulfur is found in proteins, but you don't ever find sulfur in a DNA molecule. So they took a phage that had a radioactive protein and they infected a bacterium with that phage. You can see that here. Then, after the bacteria cells were infected, they put the mixture into a blender. And so they basically took the empty protein parts that are here and ripped them off of the surface of the bacterium. Then they were able to put the liquid into something called a centrifuge, and the centrifuge separated things out by their density. And so at the top of the centrifuge tube, there's a liquid that contained the outside virus coats from the viruses, and at the bottom is a pellet of bacteria cells that contain bacteria, and inside of the bacteria is the DNA that is not radioactive. Then they repeated their experiment with radioactive DNA. So the radioactive DNA is shown here, and you can see that the virus injects the DNA into the cell while the virus coat stays on the outside of the cell. And then they did the same thing. The infected cells were put into a blender, and then in the blender, the virus coats were again ripped off of the surface of the cell, and then the DNA stayed inside of the cell. And so then they were able to again look at the liquid. The DNA went to the pellet at the bottom. The virus coat stayed in the liquid at the top, and they were able to detect radioactivity in the bacteria but not the liquid. And so they knew that the genetic part of the virus had to be the DNA because that's what went inside of the cell to change it. So here we can do a recap. Hershey and Chase use a T2 phage, which is the virus that infects bacteria. In one of the T2 phages, they put radioactive sulfur. That's in proteins. And when they did that, they discovered there was no radiation inside of the bacteria. Then they repeated their experiment with radioactive phosphorus, and they discovered that when they did that, they found the phosphorus inside of the bacteria, meaning that the DNA went inside of the bacteria. So this proved that the DNA must be the part of the virus that contains the genetic material and not the protein coat that stays outside of the bacteria. Another scientist who made a big contribution was Rosalind Franklin. She's a scientist who took really, really good X-ray diffraction images of DNA, and you can see the picture that she took here. That picture is what helped the next set of scientists called Watson and Crick determine the molecular structure of DNA. Her picture was given to Watson and Crick without her knowing by Morris Wilkins. And so he used her picture or shared her picture rather, without telling her. But this picture was really important because of the pattern that it shows. It helped determine the positions of the different atoms in the DNA molecule, and they, from this image, learned that DNA must be a helix. Using the image that Franklin had taken, these two scientists, Watson and Crick, are the ones who figured out the final shape of DNA as a double helix, meaning that it has two strands that are connected to each other and they have a twisting shape. So now we know the structure of the DNA molecule. DNA stands for deoxyribonucleic acid. You can break that word down into 3 parts, right? So there's the D for deoxy, and then the nucleic, and then acid is the final word. So deoxyribonucleic acid is what DNA stands for. The shape of the DNA molecule, again, is that double helix. So it's got 2 strands that twist around one another. And it's made of 3 different types of molecules. The 3 molecules that are used in a DNA molecule make up a nucleotide. So the 3 parts are a 5-carbon sugar. In DNA, it's called a deoxyribose sugar. And then another part of the DNA molecule is something called a phosphate. And there's also a structure called a nitrogen base. And there's 4 different types of nitrogen bases. So you can see over here, we have the 5-carbon sugar, which is here. And then we have the phosphate, which is this molecule. And then we have the nitrogen-containing base is this molecule. The 4 different nitrogen-containing bases mean that we have 4 different kinds of nucleotides found in a DNA molecule. So those bases include the purines, which have 2 rings. So there's adenine, and that's this molecule here. And then we also have guanine, that's this molecule here. So those are both called purines. The other type of nitrogen base is called a pyrimidine. They only have 1 ring, and those are thymine, which is here, and cytosine, which is here. And when they are in a DNA sequence, remember A, or adenine, which is represented by an A, always matches up with T, and G, which is guanine, always matches up with C. When DNA has those base pairs, as they're called, again, we just learned that A and T always match up and G and C always match up. This picture shows how they match up. So when you have a thymine and an adenine, they always pair up with each other with 2 hydrogen bonds. Remember, a hydrogen bond is a weak force that attracts 2 atoms to one another. When we move to cytosine and guanine, which we can see here, they match up with each other with 3 hydrogen bonds. And that's how the A's and T's always know how to find each other, and the C's and G's always know how to find each other. So to recap, we know that DNA is a double-stranded molecule that looks like a double helix. We also know that the strands are complementary, which means that on one side you'd have an A and the other side you'd have a T, for example. They're not identical to each other. We also know that they're anti-parallel, meaning that if one strand is running this direction, the strand that goes across from it is running in this direction. And to review, remember A always matches with T and G always matches with C. And we have learned since that we discovered the structure of DNA that humans have 3 billion base pairs in their DNA. So you have many, many DNA molecules in all of your cells right now.